CSIR Evolution Chapter 4. Chapter 4 History of Earth. To understand the long-term patterns of evolutionary change, we must think in time frames spanning many millions of years and imagine events and conditions of Earth that were very different from those we now observe. The Earth of the distant past was almost like a foreign planet inhabited by strange organisms. The continents were not where they are today, and climates were sometimes dramatically different from those of today. One of the remarkable achievements of 20th century science has been the development of sophisticated techniques using rates of decay of various radioisotopes, changes in Earth's magnetic field, and the presence or absence of certain molecules for inferring past conditions and dating them accurately. 41. Determining age. It is difficult to age rocks because any type of rock could have been formed at any time during Earth's history. It is easier to determine the ages of rocks relative to one another. The first person to recognize that this could be done was the 17th century Danish physician Nicholas Steno. Steno realized that in undisturbed sedimentary rock, the oldest layers, or strata, lie at the bottom and successively higher strata are progressively younger. Geologists subsequently combined Steno's insight with their observations of fossils, preserved remains of ancient organisms contained within the rocks. They discovered that fossils of similar organisms were found in widely separated places on Earth, that certain organisms were always found in younger rocks than others, and that organisms in more recent strata were more similar to modern organisms than were those found in lower, more ancient strata. With this information, they learned much about the relative ages of sedimentary rocks and patterns in the evolution of life. But they could not tell how old the rocks were. 4.1.1 Radiodating of rocks. Radioactive isotopes decay in a predictable pattern over long time periods. During each successive time interval, an equal fraction of the remaining radioactive material of any radioisotope decays, either changing to another element or becoming the stable isotope of the same element. For example, in 14.3 days, 1/2 of any sample of phosphorus 32p decays to its stable isotope, phosphorus 31p. During the next 14.3 days, 1/2 of the remaining half decays, leaving 1/4 of the original 32p. After 42.9 days, 3 half-lives have passed, so 1/8, that is, 1/2x1/2x1/2 equals 1/8, of the original 32p remains. Each radioisotope has a characteristic half-life. Tritium, 3H, has a half-life of 12.3 years, and carbon-14, 14C, has a half-life of about 5,700 years. The half-life of potassium-40 of 4K is 1.3 billion years. That of uranium-238, 238U, is about 4.5 billion years. To use a radioisotope to date a past event, we must know or estimate the concentration of the isotope at the time of that event. In the case of carbon, we know that the production of new 14C in the upper atmosphere, by the reaction of neutrons with 14N, just balances the natural radioactive decay of 14C. Therefore, the ratio of 14C to its stable isotope, 12C, is relatively constant in living organisms and their environment. However, as soon as an organism dies, it ceases to exchange carbon compounds with its environment. Its decaying 14C is no longer replenished, and the ratio of 14C to 12C in its remains decreases through time. The ratio of 14C to 12C in fossil organisms can be used to date fossils related to human, and thus the sedimentary rocks that contain those fossils that are less than 50,000 years old with a fair degree of certainty. 4.1.2 Indirect estimation of age of sedimentary rocks. Fossils are generally found in sedimentary rock, not igneous rock. Sedimentary rocks can be dated using radioactive carbon, but because carbon decays relatively quickly, this only works for rocks younger than about 50,000 years. So in order to date older fossils, scientists look for layers of igneous rock or volcanic ash above and below the fossil. When molten rock cools, forming what are called igneous rocks, radioactive atoms are trapped inside. Afterwards they decay at a predictable rate. By measuring the quantity of unstable atoms left in a rock and comparing it to the quantity of stable daughter atoms in the rock, scientists can estimate the amount of time that has passed since that rock formed. Scientists date igneous rock using elements that are slow to decay, such as uranium and potassium. By dating these surrounding layers, they can figure out the youngest and oldest that the fossil might be. This is known as bracketing the age of the sedimentary layer in which the fossils occur. The decay of potassium-40 or uranium-235 is most commonly used to date most of the ancient events in the evolution of life, such as fossil remains of more than 100 million-year-old dinosaur bones. Radioisotope dating, combined with fossils, is the most powerful method of determining the ages of rocks. But there are many places where sedimentary rocks do not contain suitable volcanic intrusions and few fossils are present. In these areas, other dating methods must be used. One method, known as paleomagnetic dating, is based on the fact that Earth's magnetic poles move and, occasionally, reverse themselves. Because both sedimentary and volcanic rocks preserve a record of Earth's magnetic field at the time they were formed, paleomagnetism helps determine the ages of those rocks. Other dating methods use continental drift, sea level changes, and molecular clocks. Using these methods, geologists have divided the history of life into eras, which in turn are subdivided into periods, Table 3. The boundaries between these divisions are based on major differences in the fossil organisms contained in successive layers of rocks. The divisions were established before the ages of the eras and periods were known. 4.2 The changing face of Earth. Since its origin, Earth has undergone many physical changes that have influenced the evolution of life. 4.2.1 Changes in position of continents and sea level. The maps and globes may give an idea of a static Earth. We might think that the continents have always been where they are. But we would be wrong. Earth's crust consists of a number of solid plates approximately 40 kilometers thick, which float on a fluid mantle. The mantle fluid circulates because heat produced by radioactive decay sets up convection patterns in the fluid. The plates move because material from the mantle rises and pushes them aside. Where plates are pushed together, either they move sideways past each other, or one plate moves under the other, pushing up mountain ranges. The movement of the plates and the continents they contain is known as continental drift. At times, the drifting of the plates brought the continents together, at other times, they drifted apart. The positions and sizes of the continents influence ocean circulation patterns, sea levels, and global climate patterns. Mass extinctions of species, particularly marine organisms, have usually accompanied major drops in sea level, which exposed vast areas of the continental shelves, killing the marine organisms that lived in the shallow seas that had covered them. Figure 23. Figure 23, sea levels have changed continuously. Most mass extinction, indicated by ***, of marine organisms have coincided with period of low sea levels. 4.2.2 Continental Drift Hypothesis. This theory was proposed by Alfred Wegener in 1912. The history of Earth helps explain the current geographical distribution of species. For example, the rise of volcanic islands such as the Galapagos creates new environments for founders that had reached, and adaptive radiation fills many of the available niches with new species. On a global scale, the drifting of continents is the major geographical factor correlated with the spatial distribution of life. The continents are not fixed, but drift about Earth's surface like passengers on great plates of crust floating on the hot, underlying mantle, figure 24. Unless two land masses are embedded in the same plate, their positions relative to each other change. For example, North America and Europe are presently drifting apart at a rate of about 2 cm per year. Many important geological processes, including mountain building, volcanism and earthquakes, occur at plate boundaries, called as subduction zone, figure 25. Figure 24, the modern continents on Earth's crust plates. The arrowheads indicate zones of tectonics events. Figure 25, at subduction zone, where plates move towards each other, the denser plate dives below the less dense one, creating a trench. The abrasion at subduction zones causes earthquakes, volcanic activities, and mountain formation. Two events related with continental drift had an especially strong influence on life. The first event about 250 million years ago, near the end of Paleozoic era, plate movements brolite all the land mass together to form a supercontinent that has been named as Pangaea. Figure 26. This event has strong effect on life. Species that had been evolving in isolation came together and competed. When the land masses coalesced, the total amount of shoreline was reduced, and there is evidence then pace that ocean basins increased in depth, which lowered sea level and drained the shallow coastal seas. Then, as now most marine species inhabited shallow waters, and the formation of Pangaea destroyed a considerable amount of that habitat. The continental interior, which has a drier and more erratic climate than coastal regions, increased in area substantially when the landmass came together. Changing ocean currents also would have affected land life as well as sea life. The formation of Pangaea surely had a tremendous environmental impact that reshaped biological diversity by causing extinctions and providing new opportunities for taxonomic groups that survived the crisis. Figure 26, the history of continental drift. The second dramatic event in the history of continental drift was around 180 million years ago, during the Mesozoic era. Pangaea began to break up, causing geographical isolation of land mass. As the continents drifted apart, each became a separate evolutionary arena, and the fauna and flora of the different biogeographical realms diverged. The separation pattern of continents solves many biological puzzles. For example, paleontologists have discovered similar matching fossils of Triassic reptiles in Ghana, West Africa, and Brazil, South America. These two parts of the world, now separated by 3,000 km of ocean, were contiguous during the early Mesozoic era. 4.2.3 Evidences in favor of continental drift hypothesis. Wegener's hypothesis about continental drift was based on several lines of evidence. The first was the remarkable fit of the South American and African continents as part of Gondwanaland, shown in Figure 27. Furthermore, Wegener noted the occurrences of matching plant and animal fossils in South America, Africa, India, Antarctica, and Australia was best explained by continental drift. Many of these fossils were of large land animals, such as the Triassic reptiles Lystrosaurus and Cynognathus that could not have easily dispersed among continents, or of plants whose seeds were not likely to be dispersed far by wind, such as the fossil fern Glossopteris. Figure 27, the fossil remains of dinosaurs and ancient plants are spread across regions of South America, Africa, India, Antarctica, and Australia, which were once united as Gondwanaland. Also, the discovery of abundant fossils in Antarctica was proof that this presently frozen land must have been situated much closer to temperate areas in earlier geological times. The distribution of the essentially flightless bird family the ratites in the southern hemisphere is also the result of continental drift. The common ancestor of these birds occurred in Gondwanaland. As Gondwanaland split apart, genera evolved separately in each continent so that today we have ostriches in Africa, emus in Australia, and rheas in South America. Figure 28, present-day, dark, and fossil distributions, light shading, of Nothofagus trees. Similarly, the southern beech trees of the genus Nothofagus have widely separate distributions in the southern hemisphere, Figure 28. The southern beechnuts have limited powers of dispersal and are not thought to be capable of germinating after long-distance ocean travel. However, trees produce abundant pollen that is found in many fossil records throughout Gondwanaland. Taken together, present-day and fossil records suggest a Gondwanaland ancestor. Continental drift is not the only mechanism that creates disjunct distributions. The distributions of many present-day species are relics of once much broader distributions. For example, there are currently 4 living species of a tapir, 3 in Central and South America and 1 in Malaysia. Fossil records reveal a much more widespread distribution over much of Europe, Asia, and North America. The oldest fossils come from Europe, making it likely that this was the center of origin of tapirs. Dispersal resulted in a more widespread distribution. Cooling climate resulted in the demise of tapirs in all areas except the tropical locations. Another well-known example of a disjunct distribution is the restricted distribution of monotremes and marsupials. These animals were once plentiful all over North America and Europe. They spread into the rest of the world, including South America and Australia, at the end of the Cretaceous period when, although the continents were separated, land bridges existed between them. Later, placental mammals evolved in North America and displaced the marsupials there, apart from a few species such as the opossum. However, placental mammals could not invade Australia because the land bridge by then was broken. The elephantidae and camelidae also have disjunct distributions. Elephants evolved in Africa and subsequently dispersed on foot through Eurasia and across the Bering Land Bridge from Siberia to North America, where many are found as fossils. They subsequently became extinct everywhere except Africa and India. Camels evolved in North America and made the reverse trek across the Bering Bridge into Eurasia. They also crossed into South America via the Central American Isthmus. They have since become extinct everywhere except Asia, North Africa, and South America. 4.2.4 Biogeographical realms. Alfred Russel Wallace was one of the earliest scientists to realize that certain plant and animal taxa were restricted to certain geographic areas of the Earth, called biogeographic realms. For example, the distribution patterns of guinea pigs, anteaters, and many other groups are confined to Central and South America, from central Mexico southward. The whole area was distinct enough for Wallace to proclaim it the Neotropical realm. Wallace went on to divide the world's biota into 6 major realms, or zoogeographic regions: Nearctic, Palearctic, Neotropical, Ethiopian, Oriental, and Australian. Figure 29. These regions are still widely accepted today, though debate continues about the exact location of the boundary lines. Biogeographical realms correspond largely to continents, but more exactly to areas bounded by major barriers to dispersal, like the Himalayas and the Sahara Desert. Within these realms, areas of similar climates are often inhabited by species with similar appearance and habits, but from different taxonomic groups. Figure 29. The zoogeographic regions recognized by A.R. Wallace. For example, the kangaroo rats of North American deserts, the jerboas of Central Asian deserts, and the hopping mice of Australian deserts look similar and occupy similar hot, arid environments, but they arise from different lineages, belonging to the families Heteromyidae, Dipodidae, and Muridae, respectively. This phenomenon, called convergent evolution, has led to the emergence in each realm of herbivores and predators which have evolved from different taxonomic ancestors. In some cases, individuals have been able to disperse from the area where the group originally evolved. This kind of dispersal is obviously easier for birds and insects, which have the power of flight, or for aquatic organisms, which can drift with the tide. Cattle egrets, Bubulcus ibis, crossed the South Atlantic from Africa to northern South America in the late 1800s, possibly with the assistance of humans. Since then, they have spread widely in South America and the United States. In 1987, a bald eagle was found on the southern tip of Ireland, having presumably crossed the Atlantic in the other direction, from the United States. 1.3. Change in Earth's atmosphere. The continents have moved irregularly over Earth's surface, but some physical changes on Earth have been unidirectional. 3.1 Changes in oxygen concentration. The atmosphere of early Earth probably contained little or no free oxygen, O2. Oxygen concentrations in the atmosphere began to increase markedly about 2.5 billion years ago, when certain bacteria evolved the ability to use water as the Source of hydrogen ions for photosynthesis. By chemically splitting H2O, 2H2O4H plus O2 plus 4E, these bacteria generated atmospheric O2. As a waste product, in addition, they made electrons available for reducing CO2 to form organic compounds. One lineage of oxygen-generating bacteria evolved into the cyanobacteria. These ancient photosynthesizers are abundantly preserved in the fossil record. Cyanobacteria liberated enough O2 to open the way for the evolution of oxidation reactions as the energy source for the synthesis of ATP. Their ability to split water doubtless contributed to their extraordinary success. The evolution of life irrevocably changed the physical nature of the planet. Living organisms not only added O2 to Earth's atmosphere, but also removed most of the CO2 from the atmosphere by taking it up and transferring it to ocean sediments with their remains when they died. When it first appeared, oxygen was poisonous to the anaerobic prokaryotes that inhabited Earth at the time. Those prokaryotes that evolved the ability to metabolize O2 not only survived, but also gained a number of advantages. Aerobic, oxygen-using, metabolism proceeds at higher rates and can extract more energy from compounds than the anaerobic metabolism prevalent among living things until then. Consequently, organisms with aerobic metabolism have replaced anaerobes in most of Earth's environments. An atmosphere rich in O2 also made possible larger cells and more complex organisms. Small, unicellular aquatic organisms can obtain enough O2 by simple diffusion even when O2 concentrations are very low. Larger unicellular organisms have lower surface area to volume ratios. In order to obtain enough O2 by simple diffusion, they must live in an environment with a relatively high concentration of OZ bacteria. Can thrive on 1% of the current atmospheric OZ levels, but eukaryotic cells require oxygen levels that are at least 2 to 3% of current atmospheric concentrations. About 1.5 billion years ago, O2 concentrations became high enough for large eukaryotic cells to flourish and diversify, figure 30. Further increases in V atmospheric O_2 levels 700 to 570 Maya enabled multicellular organisms to evolve. The fact that it took many millions of years for Earth to develop an oxygenated atmosphere probably explains why only unicellular prokaryotes lived on Earth for more than 1 billion years. Figure 30, as oxygen concentration increased, the size of cells and organisms increased. In contrast to this largely unidirectional change in atmospheric O2 concentration, most physical conditions on Earth have oscillated in response to the planet's internal processes, such as volcanic activity and continental drift. External events, such as collisions with meteorites, have also left their mark. In some cases, these events caused mass extinctions, exterminating a large proportion of the species living at the time. 4.3.2 Changes in temperature and moisture. Through much of its history, Earth's climate was considerably warmer than it is today, and temperatures decreased more gradually toward the poles. At other times, however, Earth was colder than it is today. Large areas were covered with glaciers during the end of the Precambrian and during the Carboniferous, Permian, and Quaternary periods, but these cold periods were separated by long periods of milder climates, figure 31. Because we are living in one of the colder periods in the history of Earth, it is difficult for us to imagine the mild climates that were found at high latitudes during much of the history of life. Weather often changes rapidly, climates usually change slowly. However, major climatic shifts have taken place over periods as short as 5,000 to 10,000 years, primarily as a result of changes in Earth's orbit around the Sun. A few climatic shifts appear to have been even more rapid. For example, during one quaternary interglacial period, the Antarctic Ocean changed from being ice-covered to being nearly ice-free in less than 100 years. Such rapid changes are usually caused by sudden shifts in ocean currents. Climates have sometimes changed rapidly enough that extinctions caused by them appear instantaneous in the fossil record. Figure 31, hot/humid, H/H, and cold/dry, C/D, conditions have alternated over Earth's history. 4.3.2 Influence of volcanoes on Earth life forms. Most volcanic eruptions produce only local or short-lived effects, but a few very large. Volcanic eruptions have had major consequences for life. The collision of continents during the late Permian period, about 275 Maya, to form. A single, gigantic land mass, called Pangaea, caused massive volcanic eruptions. The ash the volcanoes ejected into the atmosphere reduced the penetration of sunlight to Earth's surface, lowering temperatures, reducing photosynthesis, and triggering massive glaciation. Massive volcanic eruptions also occurred as the continents drifted apart during the late Triassic period and at the end of the Cretaceous. 4.3.5 External events triggering changes on Earth. At least 30 meteorites between the sizes of baseballs and soccer balls hit Earth each year. Collisions with large meteorites are rare, but large meteorites have been responsible for several mass extinctions. Evidence for these collisions is found in the craters that resulted from their impact, dramatic disfigurations of rocks, microspherules, and shocked quartz crystals, and within giant molecules that contain trapped helium and argon with isotopic ratios characteristic of meteorites, which are very different from the ratios found on Earth. Also, fern fossils are abundant in rocks that formed at the end of the Triassic and Cretaceous periods. Because ferns can more quickly colonize and survive in bare environments than most other plants, their abundance suggests that meteorite impacts had scoured vast areas of Earth's surface. The first extraterrestrial impact to be documented was that of a meteorite about 10 kilometers in diameter that caused a mass extinction at the end of the Cretaceous period, about 65 Maya. The first clue that a meteorite was responsible came from the abnormally high concentrations of the element iridium in a thin layer separating rocks deposited during the Cretaceous from those deposited during the Tertiary. Iridium is abundant in some meteorites but is exceedingly rare on Earth's surface. The force of the impact ignited massive fires, created great tidal waves, and sent up an immense dust cloud that blocked the sun, thus cooling the planet. As it settled, the dust formed the iridium-rich layer.